So I'm going to follow up today on the secondary structure prediction, and let me just begin with a brief recap. Protein sequences fold into unique three-dimensional structures. The 3D detail in the 3D structures relate to the function. We can infer the similarity of two proteins by sequence alignments. There's sequence, there's sequence profile, there's profile. Go further down into the twilight zone. Experimentally determining a three-dimensional structure for a protein takes over $100,000, sometimes much over it, so it can be $1 million, but many sort of take in that ballpark. Today in the PDB we have about 120,000. Most of those come from X-ray crystallography. X-ray crystallography shown here in CERN is done by very large X-ray beams, here spanning over 5 kilometers. Once you have structures, you can apply comparative modeling. Comparative modeling uses the idea that two proteins with very similar sequences have similar structure. You have a protein of known structure, you have a protein of unknown structure, they are similar in sequence. You simply read off the coordinates of the one with known structure because these two are similar. This way, we can essentially do about 40 million different models. At 100,000 per protein, this translates to 4 trillion. And this is more than the GDP of France and England. To give you one example. That's the good news. The bad news is most of these 40 million models that roughly is one model in every other protein, most of those do not span the entire protein. Most of those span relatively short fragments in these proteins. In fact, for most residues, we cannot apply comparative modeling. That's the downside. Comparative modeling gets 3D structures. 2D predictions we will get to in the future. But for everything else, for which we cannot do comparative modeling, essentially all we can do is 1D prediction. Secondary structure is 1D strings, alpha helices, beta strands. And we get to this slide. Are there any questions? So this was sort of a recap of the lecture until this point. Any questions? So secondary structure, the background, called secondary structure, often abbreviated this way, often called 2D prediction. Is that right?
No.
Let me repeat. 3D, 2D contacts, secondary structure is a string. Yes, helices are formed by hydrogen bond formation. Those are contacts. But ultimately, the entire thing that we're trying to predict here is a 1D string. So secondary structure in the sense in which it's predicted is absolutely not 2D. It's a total misnomer, which you find a lot in books. Now, I try to use this slide for classification. So when proteins are very similar in sequence, close homology, we know they have similar function, they have similar structure, they have similar everything, and we can use that. Then for the blue ones here, we have some sort of more distant similarity. We may not be able to get the blue ones from sequence alignments alone, but maybe from profile alignments we would sort of get the blue ones here. But ultimately, this is the reach of comparative modeling, red and blue. And we want to predict secondary structure, we want to predict it for actually what is green and orange here. We may never reach with machine learning tools or any prediction methods, we will never reach the totally green ones. But somehow machine learning sort of expands, gets out. That's one way of looking at it. By the way, getting back to the comparative modeling coverage, bless you, on the percentage residue, I said most residues cannot be modeled. Here's another way of showing that. And for the remainder here, we can only do something where we predict, for instance, secondary structure. DSSP, so when I take the protein database of 120,000 known structures, or read off the 3D coordinates secondary structure, I use the tool DSSP. DSSP uses the hydrogen bond patterns to define sort of 3 types of states that are helix. 2 that relate to strands, typically abbreviated with E. So that's typically abbreviated with H, and everything else that is not well defined. Initially, when the authors of DSSP developed it, they looked in the database and they found that there are particular peptides that are always found in the helix, some always found in the beta strand, some always found in the other state, and some are found Here they published a few pentapeptides, 3 shown here, that are found in alpha helix and beta strand. How can that be the case? How can you have a peptide of 5 residues that exists, the same 5 residues, in one protein there in an alpha helix and another protein there in a beta strand? Bless you.
Thank you.
Well, I guess if they're at the beginning, let's say, of the sequence.
That's a great idea. However, this is a great idea, Gary, but many proteins, also very often, so the beginning of a protein is statistically more than any other place in this state, in the non-regular state, in other. But could be the reason. Something else? If they were not in the beginning, if they were somewhere in the middle, both. What could be? Is it trivial? Are you guys thinking and it's too warm to sort of operate the machinery, or the bulbs are, yeah?
I'm pretty sure that there are actually some sequences that can be both, depending on the situation.
Exactly, it's the environment. So there is chameleon. What do you know about that?
I heard about this in another lecture.
Okay. Yes, we will get back to that. So this is sort of the simplest example of a chameleon example. So since the formation of a beta strand is, for instance, not completely local, maybe if they had other global connections, they could be helix in some situation and beta strand in another. She refers to one particular example where this actually happened for 11 consecutive residues. That happens to be called chameleon. Okay, let's go into simple solutions for secondary structure prediction. The way we do that, we take known structures, find the way the SSP did that here, find the longest stretch that is always in a helix, find the longest stretch that is always in a strand, longest stretch that is always in a loop, and then when we have our new sequence, we simply match it to these steps that we found in the database, right? That's a relatively simple way of doing it. I will show you in a moment that the first method was even more simple than that, it was even simpler. But what is actually the problem with this method? The problem is you don't find, so you cannot predict for every single residue. Some you find, some you will not find, right? So it will not always work. Now, the first method again was, as I said, much simpler, and so we have to see here. This is the first methods that sort of postulated that secondary structure exists. St. Georgi, relatively briefly thereafter, published a paper in which he predicted secondary structure. And so St. Georgi, again, the first protein structures, Kendrew, Perutz, myoglobin and hemoglobin, 5860, and St. Georgi, 5860, I said, St. Georgi published the first secondary structure prediction method before we ever knew a structure. Okay, he actually was sort of a very remarkable man. We got a Nobel Prize in '37 for the combustion process, vitamin C related. But this secondary structure prediction method simply came from the logic of looking at the 20 amino acid, saying, well, you know, if you have a proline is a ring, it breaks the way the chain turns. When you look at the helix, and the helix stops, you need to sort of break the winding, right? So you come out of a helix, you need to have a new direction. And for that, you need a proline. That's as simple as this secondary structure prediction was. So if the proline breaks the helix, then before the proline is a helix. Whenever you find a proline, you say that's ending helix, and there are some residues before the helix, before the proline that you call helix. That's the simplest method you can have for secondary structure prediction, and you can sort of go from this one here to a little bit more complicated, going for probability of proline being in a helix, probability of all 20 amino acids to be in all states, and you simply do that by counting from the known structures. Now, the first sort of methods that did that in a larger scale are referred to as Chow-Fassman, so 2 authors, and GOR, those are 3 authors, Garnier is G, Oscarthorpe O, R, Barry Robson, So that's the biologist and that's the mathematician in this equation. And essentially, I call them the first generation of methods. What they use is an individual amino acid information. So they essentially say whether the amino acid is a valine at position I or valine at position J. The probability of forming a particular secondary structure is the same. It's completely independent. Where you sit, all these V's have the same probability of doing a particular secondary structure. Now, the first issue here is how to assess performance of such a method. That got to the issue of how to get secondary structure that led essentially to DSSP. So using Pauling's hydrogen bonding pattern in the dictionary of protein secondary structure of proteins, DSSP, to assess secondary structure prediction methods. So that's Chrysander and Wolfgang Kapsch. And again, Chrysander, I have to update that, is now at Harvard. And is one of the sort of people who started the field of bioinformatics much more than others. Immensely visible person. Anyway, so how good do they perform these methods? The first question here is, what do you measure? So here is the observed string. That's the number he is counting, first residue, second, and so forth. Observed blank means other state. E is the extended strand in blue. H is the helix in red. And what you see is the string observed and the string predicted. So, how do you measure performance? How good is that? What's the simplest way of doing it? Just throw some measures at me. Not so many. I mean,
just accuracy, how many are correctly predicted, divided by the number of residues that are predicted.
So this is typically referred to as Q3, a three-state accuracy, where you essentially ask how many in three states are correctly predicted. So you predict the three states, helix, strand, other, and you simply ask how many of those are right. So it's like an edit distance, right? No, it's not an edit distance, but you essentially count whenever there's the same symbol between the two. And by the symbol, so I actually published let's get back to this point here. The method published a level of 63% accuracy in this Q3 measure. Is 63% a high number?
That's better than random.
What is random?
Well, in this case,
What is it? Somebody else. What is random here? So the statement is 63 is better than random. Before we can say whether that statement is correct or not, we have to figure out what is random. So what is random?
50%, isn't it?
This is an answer, but you sort of have a question in your answer.
Yeah, I'm not sure, but I think it should be.
Should be. So somebody can argue for 50. Somebody else. Who wants to argue for 50 or who wants to argue for a different number?
I'm not sure if that's right. We have 3 residues, right? I'm saying
There are many people in the room who are not sure whether that's right.
3
But what is right? Argue.
bases.
You have 3 states. So is 50 a good number?
So the chance of getting the right one at random would be 1 out of 3.
Right. In principle, if you had them equally distributed. Right? So now we have to sort of, if the 3 classes, if 30% of the residues would be helix, 30% strand, and 30% other, then the answer would be random is 33. Since the distribution is not equal, and if you then sort of still do it randomly, not predict the majority class, then the number goes up a little bit. It's ballpark 30, 36. Okay, so 36, 63, clearly better than random, but much less clear. That brings us to another point. What is the ceiling? Meaning, so the lowest level, the floor, so to speak, the bottom, is 36. What's the top you can achieve? It's 100%. So one way you can think about this is say 100. My question to you, I'm telling you 100% is not the right answer. Why not? How can you argue against I'm arguing that 100% cannot be your objective goal. You will never achieve that. Why? What do you know about secondary structure formation, or what do you know about the problem here that will explain that? Proteins move. You may have all seen, I've shown you some examples for NMR models. Somebody in the room had seen them before, immediately guessed that different solutions are different NMR models. So, in fact, movement can change, it does change secondary structure. Yes, there are regions of secondary structure where it doesn't change. But it's also true that in almost every single protein, the movement over time may change the ends of some helices or strands. Okay? That fraction, you cannot predict better than you can observe it. You observe at two different time points, because you observe slightly two different motion states, you get a different result in terms of secondary structure. And that level, you cannot be more accurate than that, because there is no frozen right. What you would best, if you had the optimal prediction methods, you would get this sort of difference, right? You would get a level that is close to the difference between what you see. And incidentally, maybe we get back to that, I can't remember. But let me just put down the number. It's in the ballpark of 90. And I may or may not go back to that number. Now, when they did DSSP, they found out that in fact the number is lower. When that was reassessed in 94, so the number looked much more like 50 to maybe 55. So this still is clearly better than 36. So even using a single residue statistics or probability is giving you a secondary structure prediction method that does outperform random, which you all know in computational biology is not always the case. So there are tons of things out there that do worse than random. And I'll show you one of those things here in context of secondary structure prediction in a moment. Okay, so we want to do better. And since we have a first generation method, there's a second generation method, obviously. Any idea what the second generation does to do better? We discussed it on Tuesday.
Not only single residues. Not only single residues.
Not only single residues, we expand. So essentially, we begin to say, okay, these two valents here have a different role, because one is when I look at the segment here of whatever, 11 or 13, in this case 11 consecutive residues in green. When I look at these 11 residues, this one valent is at the beginning, this is at the end, and this is in the middle, and they have different roles in this segment. These valents are next to each other, this is a little bit further apart, and all of that may matter if I wanted to answer whether that valent here is an beta strand or not. Okay, so use that information. And the initial one, again, I briefly mentioned, that was the segment 83 method from Kapsch-Zender. That was the original reason why they developed DSSP. The next level, or the more rigorous way of doing it, goes back to Barry Robson here, who essentially worked out the conditional probability model for, and what I show here is a window of 7 consecutive residues. So I'm essentially looking at the conditional probability, given that this one here is, say, a P, a proline, that is next to two away from a valene in this direction, 2 away from a valene in that direction. What's then the probability that it breaks a helix? So he built up these conditional probabilities, he worked this out. It in fact sounds very easy. To write it down is not that trivial, and that was sort of the brilliance that went into a method that was referred to as goal 3, leading to levels of performance accuracy around 55 to 60. So the problems with this sort of method, the first problem is not 100%. Okay, they already told you 100% we cannot reach there. There's dynamical movement that will change the ceiling here. But ultimately, the argument then was the maximal number that we can reach was 65%. How do you believe they came to this number? Let's call it is something about social behavior of scientists, this question, more than logic. So you will find many books, or even textbooks, where there was a clear case, it's a clear statement, secondary structure cannot be predicted at levels of higher than 65%. And they give reasons. But how do you believe they got to this number? That's very simple. There was no method. Everybody tried to develop a method. Nobody reached it higher than 65, so they said 65 is the limit. In fact, at that point in the beginning of the 90s, that's when I started, and for my PhD, at some point I counted there were 400 different publications within a few years, 400 different algorithms applied to this problem. So essentially anything that you can possibly think about was applied to predict secondary structure. People really tried everything, and they always remained sort of at a magical level that was in the ballpark of, actually something that was in the ballpark of 60, not 65. But they always claimed 65, so. Now, ultimately, the reason what people argued is because some of the formation of secondary structure is global. And that's back to the pentapeptide story that I talked about, to the chameleon story. So ultimately, helices, yes, they are. The hydrogen bonds in the helix is local, but not all structure formation is as local as a helix. In fact, when we look at how well do methods predict beta strands, and what I'm showing here now is not the Q3, but what I'm showing here is how many of the beta strands are correctly predicted. So I'm only counting the E's, right, that I predict, and I ask how many of the E's that I predict are correctly predicted, and then the answer is this is ballpark 40%. So only counting E's, now we're getting to the point where random is 50. So I do E against the rest of the universe, and I get a number that is lower than random. And why is that? People argued exactly because the beta strand formation is less local. So when I take 7 consecutive or the 6 consecutive residues here shown in the beta strand, I will not, in the window of 7 or something, will not see the hydrogen binding partner. In the helix I will. So the 2 bonding partners, in the window of 7 consecutive residues, the 2 bonding partners are there. In the beta strand, may well be not the case. This is the first strand in the protein here. This is the last strand. So in between there can be many residues, right? There's another problem that the traditional secondary structure prediction methods had, and that's the problem of short segments. What I mean is illustrated here. Sequence the actually observed secondary structure. For this case, there's an issue here with the short E's. Let's not get into that. But this is a typical prediction. And you know, this typical prediction averages out, in this particular case, I can't remember, but to a number that is most likely here higher than 60, simply because of the matching blanks. So many blanks that are placed correctly, that dominates the count here. There are also many E's that are right. And there are even some H's that are right. The problem with the prediction here is, since you do know that, let's pick that example, helices of 2 you don't have. Okay, so does that mean that here's no helix, so that this is actually blank? Or does it mean that this is actually a long beta strand? Beta strands typically are 5 residues long, so you know, you just do the H, you flip the H to an E, and you have a typical beta strand. Is that what you should do? The same issue here, but in this particular case, actually should you now switch it to a helix? Because here you have sort of dominant helices. So the problem of short segment prediction is, how do you use it in practice? So it makes sort of sense, but what you know is not right, and an expert cannot easily find how to correct, how to go from what I have to what is sort of an expert filtered version of what I have. So why not apply machine learning? And that was sort of the story at the 90s. When the 90s began as a new method to apply to secondary structure prediction, machine learning devices. And the one that I already briefly talked about on Tuesday are neural networks, where again we have an input, we have connections here, we have an output. The way the algorithm works is you multiply the value of the input with this connection. That's what comes in here. Ultimately what you do then is you have an input vector, 1, 1, J1, J11 and J12 vector of the connections here. So you multiply 2 vectors, get a dot product. Then that is sort of, you essentially sum this times this times this, sum it. That's what comes in here. You apply a sigmoid type of function, where if the sum is very highly negative, so the connections can be negative. If the sum is very highly negative, you switch to 0. It's very highly positive, you switch to 1, and something in between. And the way this sort of trains is if you had, I'm sorry, it's very faint here. There's an open circle, there's a dark circle here, and you want to classify these two circles. So the input for this one is 1 and 0. And so you train that system by changing the connections. The mistake that you do is what you get out here by what you want squared. You train it by sort of going downhill in the error space of the connections. Example here, say I had initialized my connections with 1s, which absolutely typically you don't do. You choose them randomly. But so in this particular case, you have 1 times 1, 0 times 1, you add that up is 1. And then you do the tangents, the hyperbolical tangents, so you get a value that is on the positive side. What you actually want is 0. How do you change that? How would the downhill arrow step work here? Sorry, wait, I made a mistake. But you all, so we have a value of 1, yeah, well, it's close to 1. I want this to be 1 and this to be 0. Is that where I'm going? Somehow I want this to be 0 and this to be 0. This doesn't make sense. There's something wrong in my example. Because of some reason that I cannot remember, I chose to reduce this value because it was too high. So I felt this should be 0. But this, I believe I made a mistake in my example. This one should be dark here. So this one I have to reduce. So since the output value is too high, the only way I can make it smaller is I reduce the connections, right? So I say move it to 0. In this particular case here, in fact, this is the mistake. I have to color this. So I increase because here I need a larger value. And then you keep going, so you adjust them. And ultimately what you do is you introduce lines in space for a neural network. What I show here is essentially 4 neural networks that all 4 classify the problem without any error. Okay? You may have heard of SVMs, support vector machines. SVMs always try to find the line that optimally separates the point. So in this particular case, that would be this one here. So for the SVM, there's only one solution. While for the neural network, all 4 are completely equal, and they will not differ with different random conditions or different initial conditions. You will actually find these 4 lines. If we had this problem, the so-called XOR problem, a straight line wouldn't do it, a curved line would do it, or 2 straight lines. And in the language of the neural network, 2 straight lines you do by adding a hidden layer. The computation is the same, so this is the connection times the input, the sum, the sigmoid function. This is essentially what comes out here, and you multiply what comes out here with this connection here. So what comes out here to this connection. You sum over this and that, and you apply another sigmoid function. Same thing as before. You do the same downhill move in the error space that we had before by defining some objective error. So the error again is what you want, or what you have minus what you want. You square, you sum over all output units. Go downhill. They go downhill. So this is typically referred to as batch learning. So you present one particular example and you move a tiny step of epsilon downhill. Okay, then you present another example, you move downhill. This one here is allowing you uphill moves, so prevents you from getting stuck in this minimum by essentially adding sort of a momentum term. When you apply neural networks in practice, you typically begin by choosing this epsilon as a high number, it's close to 1, or a half, 1/2, and this is even larger. And the more you understand how to solve your problem, the smaller you make these numbers. For the final prediction method, for the final thing that you want to publish, you want to have these 100 times smaller than you initially did, simply because you want these devices to sort of move in this landscape very slowly. When you see an image like that, describing here on the x-axis training time again is defined as how many times do I have to present all samples and have to optimize, right? And this is the accuracy. 100% is completely right. I see two curves. What could that be? What could this event be here? Overtraining, overfitting, exactly. And overtraining, overfitting has always, I do want to put the point, this is a kind of toy problem, but this is the kind of thing that we will always see. So that means the blue is essentially the training, cross-setting, training, testing, validation set. There are different words for that. And that's the training set. Now, that brings us to this issue of cross-validation. So if the pi represents all my experimental data. So for this example that I have here, I have 120,000 proteins in the PDB. I do a sequence reduce subset that is such that no pair has similar sequence. That brings us about 10,000 proteins. Protein has about 400 residues. 10,000 times 400 is about 4 million data points, right? That's the pi. Now I take those 4 million and I split them into training. We will get later to the cross-training and testing. Okay? And what I show here in real life is how testing looks and how training looks to predict secondary structure in this particular case. You see that the system can learn completely, not completely, we end up here at 90 something, but we train it to a very high level of accuracy. So the point of overfitting or overtraining is also somehow visible in the real life case of testing. But not so well, right? Because it is beginning to, if you in your thesis were to choose the point of where does overtraining start, you could pick this point here, because this is the first time it goes down. You could pick this point, because it's the highest point. You could pick this point also because it's sort of the highest deviation, or you could put some other point. But yes, there is sort of an effect, but it is not very dramatic. So clearly, in the toy kind of problem, you would only seeing the testing, you would see where to put it. Here you don't. You wouldn't even know from the blue here whether you have learned it. You need a green one at the same time, right? For this one here, you don't even need to see the green curve. You know exactly where to stop, right? My point here is, this is what fills all the textbooks about machine learning. In computational biology, it looks more like this. This is reality, right? So it's much more complicated how to stop, how to prevent overtraining than it seems. It's something that we keep talking about. It is an image that is very convincing, but it is very elusive as a goal in practice. Okay, let's get back to secondary structure prediction. So I said that the problems were we cannot quite get to 100%, 65 is the max, that beta strands have 40% sequence only 40% performance, and we have very short segments. What about the machine learning device, the neural network? So in the neural network here, the solution is the following. I have 13 consecutive residues in a protein. Now each of these yellow blurbs here stands for 20 amino acids. So each one is 20 input units. Not quite 21 because we need a spacer, right? You remember why we need a spacer. Because when the central residue, we predict the secondary structure for the central residue here, P. When that is the first in the protein, then what is above it is not an amino acid. That's why we need the spacer unit here, 21. Okay? So the input here is 13 times 21. Okay? There's 3 output units for helix strand other. And then there is some hidden layer in between. So what do you believe? Is that doing better than the simple statistics? a sort of fancy device. Machine learning versus simple statistics. So some people seem to be indicating yes, is what they believe. But the precise count of some is 1. Meaning that I'm slow. 9 people in the room have no opinion. Okay, so the answer is 62, not 42, 62. So essentially, in this particular case, believing that just putting some machine learning device, something fancy into the equation, that did not help at all. But we still light at the horizon. So here's what happened during the training of the neural network. Again, this is time. One count here is presenting all the samples once. That's percentage. We have 3 different curves here: beta strand in blue, alpha helix in red, and other state in green. Can you tell me what you observe? Please, not everybody at the same time. Okay, also getting
Well,
on.
this is the same situation that we had before, more or less.
What exactly do you mean? This is not testing versus training.
No. I mean, this is secondary structure prediction comparable to the results of Gore and the other methods. So we have a close to random performance on the strands,
Oh.
better performance on the helix.
Statement number 1. Yes. I'd like, Anton, what I'd like to do at this point typically is describe the part that you would in a paper call results. What you do is you begin to interpret. Essentially, the point is, but what you said is sort of close to each other. First, you have to observe that blue is always lower. And that implies, since blue is always lower than red and green, that implies that beta strand at any cutoff here is the lowest performer. So that in some sense appears to be a similar situation. I don't know whether it's quite random. It is the 40% that we had in the other one too. So that is statement number 1. Okay, next observation. That's one observation. Somebody else.
It seems that the training doesn't have all that much with helix and other, because they it sort of goes up and down at the beginning, and then it's just sort of pretty random apparently.
So there is no, so red and green do not quite improve. If anything, then green improves a little bit and red becomes below. Red and green sort of are on each other's shoulders or against each other in some sense, right? That is, so red loses against green, but there's some sort of balance, and they essentially don't do much over the course of what I see. But this is the next thing that is very, very important. It's very trivial, but this is when you see something like that, you have to sort of watch something. Actually, there's something happening before this curve starts, right? Because you see, when number 1, the first data point here, red and green essentially are at a level from which they will not change anymore much. But this is not random. So this is a level around 70%, right? This is nowhere near random. So something has happened before this point. So at some point of drilling down into detail, you may want to sort of get a resolution of something that in this plot you don't see. There's another thing that, at least for yourself and also for your thesis, you have to make the point that something has happened already. And you don't see that on this plot, right? And there's something else here that is very related, which is that, in fact, they have learned that, yes, there's not much change anymore. They have essentially, well, there's a little bit of change, but essentially they have learned before this curve starts. Well, we are here way, 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 way below random. There's a different way, we can see random in different ways, but clearly blue increases. So now you could argue, depending on where you cut this off, whether green really increases, whether red really goes down, but blue, you cannot argue. over the course of this curve, yes, blue is always below the other two, but blue grows a lot more than at least a factor of 1.5 or over a factor of 2, almost a factor of 3 from the first step to whatever it's the optimal step, right? So that brings us to a suspicion. How could that be caused? How can you now relate what you observe in this plot here to action? To, well, what could you try as the next step? What could that be related to? Any idea? Why would that happen? Throw some ideas at me. What could be the explanation for that?
Maybe after networks is more structures, it can like better predict data strand, but for helices it's always like a local.
So essentially what you say is that one way of using that is the task of predicting beta strands requires more samples, requires more detailed training. It's simpler to get helices than to get beta strands, so it takes longer. Yeah, that's true. Something else? Yeah?
Well, maybe they already initialized the network with data that they already know, with principles that they already know for the helices, you know, the structure.
Who's they?
The people who performed this training.
No. I know the people who performed that training. I know them all.
And that would explain why it starts at a high level.
No. So what explains it is really there's a limited resolution. This curve does not start from 0. I essentially show you one data point after 20. So I go over an order of magnitude here. I go from it happened to be 20,000 steps. This is 20,000 and this is 200,000 steps. But I do not show you what happens in the first 20,000. What I can tell you, they all started at a very, very low level.
Oh, okay.
They all started very close to random. And so for that, this was my first point. You do have to ask me or ask yourself to get more resolution. To, in fact, break it down what happened at time point 0. How was the system when we started? And that is not shown in here. This is something, again, I did this quite some years ago, over 25 years ago, and I just don't have the old plots anymore. Some data is lost. But anyway, so there's something else. What Agne and Franziska had this something that we have some competition event here. So these two are sort of taking it from each other, right? So maybe there's competition, and maybe ultimately, let's just look at it in a different way. So we have 62% accuracy. Q3 of 62%. So 62% of the residues are correctly predicted out of 3 states. When I now ask what fraction of these 62% of the residues, so what fraction of the correctly predicted residues are helix in red, are beta strand in blue, and are other in white. So the pi, the whole thing is 62, the 62% that you correctly predict, and of those, most are white. And the second highest fraction is red, and the lowest fraction is blue. That's the method. Oh, sorry, I have an animation in here. So the method at the end here, I really do not remember where I cut. At some point I cut, and the method produces exactly this pie chart. Okay? This is the pie chart of the a priori probability, so the distribution in the database. So the whole thing is all the 4 million, in those days was a smaller number, and that fraction is helix, that fraction is beta strand, and that fraction is other. Is something jumping at you?
Yes. biased by the classes.
It looks very similar, right? Actually, what you want, maybe, is this. So you want somehow a distribution that is equal in the performance, right? This is one way to formulate your wish list. And that gets us, how could we do that? How can we try to do that?
You could train the model giving an input at equal classes.
So, exactly. So usually what happens is at every single time step you presented a sample, and you present the samples at the same probability at which they are in the database, and you're going to change the probability. You're now going to essentially have one example from alpha helix, one from beta strand, and one from other. At every single time point. So you have little micro batches in which you make sure that every class is sampled equally. Which means that beta strand will be oversampled because it's the minority class, right? You will take more beta strand in your training than you actually have in your data set. But you will take them at equal probability. Okay? And when you do this, well, this is the repetition of what we got in the unbalanced way, and this is what you get in the balanced way. Exactly identical. When you get to the performance, beta strand is predicted exactly at the same level as other and helix. Now, you pay a price. The Q3 goes down because the majority class is other. You have now developed a method that is good at doing beta strands, and beta strand is a minority class, and since the Q3 is completely blind to the class, is dominated by the majority class, in terms of Q3, you're going to do worse, right? That's the price you pay. But the remarkable thing here is, remember there was this argument that beta strand formation is less local than alpha helix formation? Is that the reason why we predict beta strand less accurately? Is that the weather? Or is that a complicated question, or is it a simple question? Are you trying to say something, or are you trying to get wind?
Because of the class distribution, because like you said, there are less beta strands, and so the prediction is not so good as alpha.
So the point is that ultimately, yes, beta strand formation is less local than alpha helix formation, but this is absolutely not the reason why beta strand is predicted less accurately. We can predict beta strand at the same level of accuracy as alpha helix, right? So not every explanation that is sound is right. Here we have an example where the explanation is correct. I mean, it's true, beta strand is less local. It sounds okay to explain that this would lead to a lower prediction performance for beta strand, but it's wrong. With the same data, not doing anything else, you can force beta strand to predict at exactly the same level, which implies that the local preference is completely the same. Right? It just is an artifact of the way you train it. It's an artifact of the way the database is distributed. The great thing about machine learning device here is you could easily see that. For the Gore method, for the probability-based method, you could not sort of easily change that, because then you would have to really change the data, and you sort of would do all kinds of other things with that. But this is an example where you can not only use machine learning to sort of solve a problem, but to learn something about a problem and about an explanation. Secondary structure formation, the local preferences for beta strands and helices are completely identical, although that may not be very intuitive. Yes?
But does this imply that the reason for the inaccurate predictions is the same for all three classes?
No. No, that's not true. No.
We do not apply, so that still could be the reason why, in the case of the beta sheet, we don't get out the rest 40%.
Yes,
I'm not sure.
So this sounds like yes. So in physics, you typically say, try to come up with the explanation that assumes the least. And this is not the path you try. You make an explanation that sounded good before, try to survive by assuming some other reasons. So we have an absolute equal performance for all three classes, but there may still be sort of underneath hidden reasons why they are different. Why should there be? One thing is clear: if the reason is less local than helices would be the explanation, we would never force this. That much is clear. Let's not continue speculating. What you say is in principle true, and this is the kind of thing that people do in order to make explanations that sounded good.
Now,
survive.
What I'm trying to say is...
No, but logically you're right.
Can we now kick out the thing that data sheets are determined by the global structure versus the local structure? Can we now kick that out?
Yes, we can kick it out because that cannot be the dominant reason. Otherwise, these numbers would not be identical.
Okay.
It cannot be the dominant reason. It can, it could be a sort of higher order effect at some point. But this is what I'm saying. So we're talking about a higher order effect. On the first level, clearly it's not. Okay? And this was typically applied on the first level, judging from this. Okay, so now, so the machine learning already solved this problem. We can do much better for beta strands. Although beta strand is less local, we can predict it much better. But now the next issue here is about short segments. And the network that I showed you before essentially gets an alpha helix on average have 4 residues and a beta strand of 3, as opposed to observed beta strand about 5, alpha helix is about 11. So, meaning that the method that I showed you so far, the one that is balanced, so does beta strands better, or the one that is imbalanced, that does everything else, the non-regular class better, they both do not get the segments right. Why is that the case? Well, the reason is that essentially you take your 4 million residues, you put them in a database, and when you do the error reduction here, the gradient, the error reduction, the downhill move and the gradient downhill, then what you do is you pick at 2 consecutive time steps at time t and t plus 1 randomly from your data set. So at time t it could be residue 42, and at time t plus 1 it could be 660,000 or something like that, right? In these two you have absolutely no correlation between, well, beta strands have to have a certain length, alpha helices have to be a certain length, because they're completely different proteins. So at time point t and t plus 1, you have samples from completely different proteins. Now, you could possibly hope to augment that or go around this problem by training consecutive segments. This is what people do today in deep learning for image analysis, and in image analysis that makes sense. For secondary structure prediction that goes wrong. It's because you introduce strange correlations that lead to other artifacts, to other problems. But how could you solve this problem? How could you somehow put into the system that there is some correlation? On Tuesday we briefly discussed this idea that we could sort of try to fit it into the length distribution. So encourage that helices are longer. But then we got into the problem of this would lead to a lot of rules. We did not quite want to. Can you imagine a way in which we sort of can generically put that into a machine learning device? Just form groups and discuss. Maybe